Growing mini brains in a petri dish? I'm going to show you how it's done. Hey there, welcome to Science is You. Whether you're in grad school or just want to learn more about science, my goal is for you to feel confident and empowered navigating the world of scientific research. A really cool technology that's emerged within the past decade is the ability to grow miniature versions of brain-like organs called cerebral organoids in the lab. In this video, we're going to go through an original research paper that was key in the development of this process and explain the main points and key concepts so you too will know the science behind growing cerebral organoids. First, some background on how the human brain develops. Disclaimer, this is a brief background that summarizes the main points of brain development from the author's intro to get a better picture of what they're talking about. We're not going to cover every detail here, and we don't need to understand the paper. The brain starts out with cells that form a tube. Cells at the inner wall of the tube turn into another type of cell. Many of these cells will go on to ultimately become neurons. As they develop into more neuron-like cells, they move outward through the wall of the tube, eventually ending with the most mature neurons in the outermost layer, which will become the cerebral cortex. This tube then has some layers or zones that contain different types of cells at different stages of maturity. These zones all have long and fancy sounding names like subventricular zone or inner fiber layer, but instead of getting caught up in remembering all the names, I'd rather start with just having a picture of what's going on. There are a couple of key players that will come up again though. The cells that form the tube are called neuroectoderm, and the cells at the inside of the tube are called radial glia. In humans, there is a unique small layer of cells called outer radial glia. Mice, for example, don't have this, and overall the structure of the rodent neural tube just isn't as elaborate as in humans. This leads to the striking difference in brain size and function in humans versus rodents. This is important to note because mice and other rodents are often used to study human disease, so there's a need for something better. At the time this paper was written, some neural tissue had previously been grown and successfully maintained in the lab, but it didn't have all of the organized layers and types of cells we were talking about. In this paper, the authors developed an improved type of lab-grown brain tissue to use in the study of disease. They did this by using human pluripotent stem cells, which are cells that are capable of becoming almost any type of cell in the body. They started by making clusters of these stem cells. Once clustered together, the process of differentiation begins. Differentiation means a stem cell is starting to turn into another type of cell. A combination of chemicals, growth factors, and physical stimuli all go into determining what type of cell a stem cell will ultimately turn into. In this case, these clusters of stem cells were triggered to develop into neuroectoderm. Remember, this was the type of cell that formed the neural tube and started brain development. So it's a logical place to start to make a mini brain-like thing, right? These clusters of neuroectoderm cells were then embedded in small drops of a jello-like substance called matrigel. Matrigel contains lots of proteins that form a structure for the cells to grow into, providing physical stimuli that helps the cells develop into more complex brain-like tissues. This step was a key discovery in this paper that enabled the authors to generate more complex brain-like organoids than previous studies. The process of further cell differentiation into neural cells takes about 8 to 10 days, and 20 to 30 days for more defined brain-like regions to form. The authors reported that these cerebral organoids could grow up to 4 millimeters in diameter and survive over 10 months. However, they were missing a circulatory system that could bring nutrients and oxygen to cells at their dense center. Consequently, cells in the center of the tissues would begin to die off, limiting their size. Despite these limitations, the authors noted many regions that were similar to brain structure, at least in shape or form, including cerebral cortex, networks of fluid-filled spaces, retina, which are light-sensitive cells that aid in vision, and meninges, membranes that protect parts of the brain. But notably, all these regions weren't present in every organoid made. For example, the retinal cell regions were only observed in a few organoids. Still, all organoids had regions similar to dorsal cortex, an area at the back of the brain involved in cognitive functions. This is cool because this region differed the most between humans and rodents. They observed that these dorsal cortex neurons had characteristic long branching axon structures. They also saw spontaneous increases in calcium, suggesting some level of neural activity. Wait a second. Growing brains in petri dishes for real? Isn't this kind of not okay? Well, although they saw many similarities to human cerebral cortex, these organoids were still just not anywhere near as complex in structure as an actual human brain. But as technology progresses, could there eventually be some ethical concerns or barriers with these types of studies? What do you think? Share in the comments. Wait though, there's another part to this paper. The authors next wanted to try out applying this method of making cerebral organoids to study the disease microcephaly. Microcephaly is a condition where a brain size is smaller. This is thought to be due to mutations or changes in genes that play a role in cell division. But wait, why microcephaly? Why not a different disease? Well, theoretically, there are many diseases the authors could have chosen, but you can't test out everything in one single paper. So in this case, since they are proposing a better method of studying neurological disease, they picked a disease that couldn't be studied well with current methods like mouse experiments. In this case, the authors identified an actual patient with microcephaly. They were even able to determine the gene change causing the patient to have the disease. Picture this. Genes are regions in the DNA that are like an instruction template for making proteins. Changes in the genes, called mutations, can change the proteins they correspond to. Like if you rip a page out of an instruction manual, shortening or truncating a gene can result in a protein that can't perform its functions anymore, or maybe even isn't made at all. That's just what happened in this patient with microcephaly, who had loss of this protein with the very long name CDK5RAP2. So the authors took cells from the patient's skin and used them to make cerebral organoids. But wait, those are skin cells, not neurons. They aren't even the stem cells that the authors had originally used when they first started trying to make the organoids. True, but what's cool is just like stem cells can become lots of different types of cells, some cells found in skin can be made to turn back into stem cells under the right conditions. So the authors took a sample of the patient's skin cells, converted them in the lab back to stem cells, then used those stem cells to make cerebral organoids. Now, the authors had cerebral organoids with the patient's mutation. These patients' cerebral organoids were smaller overall, as would be expected since they had a mutation that causes microcephaly. Also, they had disorganized placement and orientation of some cell types and imbalanced populations of neurons and radial glia cells. That's interesting, but what is the point of doing all that? How does it help the patient? Well, of course this paper didn't provide a cure yet, but understanding what causes neurological diseases is the first step to finding cures. Here, they found that too many of the radial glia cells were turning into neurons as the organoids developed. This was part of why microcephaly couldn't really be studied well in mice, because mice don't have a large amount of radial glia, and they don't appear to be critical for proper mouse brain development. If you found this video interesting or helpful, please subscribe for more. Also, you can find the full citation for this paper and some further reading materials in the description. And remember,